So again, we're continuing on chapter 16, cell signaling, but rather than focusing on the general principles of cell signaling, we're going to be looking at the specifics of G protein-coupled receptors and enzyme-coupled receptors. Although I'm going to take a short detour to begin with, just looking at a topic brought up in your textbook and then giving you another resource if it's something that interests you. This is not something that would be on an exam or anything like that. Whenever you see a table, it's never something I would have you memorize. But what this is showing you is that there are substances that we can ingest or inhale that can affect cell surface receptors in our body. And some of them are used as medications, and some of them are used as recreational drugs. You can see things like nicotine, morphine, and heroin. Right? Nicotine stimulates acetylcholine-activated ion channels. Morphine and heroin stimulate G protein-coupled receptors. And then they have effects on our body like changing blood pressure or relief of pain. For me, two that stick out are the ones that I've worked with personally in a lab, capsaicin and menthol. When I was just out of college, I worked as a technician actually running human subjects through studies of capsaicin and menthol. Capsaicin gives a feeling of heat, and it's something you're familiar with if you eat a lot of hot peppers. It's the chemical that makes them hot. Menthol is in a lot of products like lip balms and things, and it stimulates cold. It's interesting that a chemical can make you feel a thermal effect. Capsaicin stimulates temperature-sensitive ion channels that produce heat, and menthol stimulates temperature-sensitive ion channels that make you feel cool. Both of them are used sometimes to relieve pain. So you may have heard of medicines like Icy Hot, creams that you can rub on your muscles, and they'll give you that feeling of heat and cold. That's because of these two chemicals, capsaicin and menthol. Actually, one of the other students in the lab worked quite a bit with capsaicin, and in those studies found that if you repeatedly expose skin, for example, on your arm to capsaicin, you'll actually become less sensitive to physical thermal heat than you were before the exposure to capsaicin. And also, if you pre-expose or co-expose someone to a wash, like a mouthwash of menthol, you'll reduce the perception of the heat from capsaicin. So there's some interesting stuff going on there. And these two really common additives, capsaicin is found within our food and then is also put into a lot of consumer products. Menthol is also added into a lot of products that you may use. And this page is a web link to an animation, an interactive, I guess you would say, that tries to show you what's going on in the brain when it's on different recreational drugs. They're using cartoon mice to show you this, so it may be somewhat silly, but they've taken the basic ideas of what drugs can do to the synapses in your brain. Again, this isn't something you need to look at, but if you have any interest, it's there. It may help in solidifying some concepts we've learned in Chapter 12 and Chapter 16. So this is the entry point to the interactive, showing these mice on a party. You hit play, it will bring you kind of in focus. It's got the psychedelic music going on here. And then you can select from the various mice affected by various substances. And zoom in to what is happening in the brains.
Inhibitory neurotransmitters called GABA are active throughout the brain. These neurotransmitters act to control neural activity along many brain pathways. When GABA binds to its receptor, the cell is less likely to fire.
So I'm not going to continue to go through this. You can hit next, and after a while you'll eventually eject the mouse, and if you want to look at another substance, you can. Again, not required, but it may be something that aids in your understanding of these pathways, so I thought I'd show it to you, and sometimes students find it interesting. Okay, from there I'm going to transition to specifics of the two pathways. First, the G protein-coupled receptors, and then receptor tyrosine kinases. So we're going to spend some time first. So here we are spending time with the G protein-coupled receptor. And we're beginning with its structure, which is unique. It has 7 transmembrane alpha helices. So each of these is kind of like a stitch through the cell membrane. 1, 2, 3, 4, 5, 6, 7. And we can see that the 5th and 6th alpha helices also are important in making up an extracellular domain that is where the ligand that activates the receptor binds, and an intracellular domain that's important for interacting with the G proteins that send the signal on to the interior of the cell. Now here we're looking a little bit closer at the interaction between the receptor and the trimeric G protein. I'm bringing the trimeric G protein back into focus here at the bottom, where we have the alpha, beta, and gamma subunits. Again, the alpha and the gamma are both attached to the membrane through a lipid link, and the alpha is the one that can interact with G proteins. And what happens is that once the receptor, the G protein coupled receptor, interacts with a signal molecule, it's activated and we see a change in shape. Right? There's the change from this kind of flatter shape in the intracellular surface to this rounder shape. We have an interaction between the G protein coupled receptor and the trimeric G protein, which promotes the dissociation of GDP from the alpha subunit. Because of the high concentration of GTP within the cell, GTP will bind to the guanosine nucleotide binding site and will often dissociate from the beta-gamma complex, and these two can separately activate downstream components. Another important aspect of cell signaling is not only activating a pathway, but inactivating the pathway. So we're going to look briefly at that as well. Here we have the activated alpha subunit and the separate active beta-gamma complex. We can see the activation of a target protein. So this is the target protein here. Alpha subunit is activating it, and it will continue to do so until the alpha subunit is inactivated. And again, it does this by hydrolysis of GTP to GDP. We've covered this in chapter 4, and there was a review of it at the beginning of this chapter. Once GTP hydrolysis happens, the complex of the alpha, beta, and gamma subunit will reform, and they are all inactive. I also have an animation of this. Sometimes these events are nice to see in kind of an active form, so I'm going to go and play that now.
Many G protein-coupled receptors have a large extracellular ligand-binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the two activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound. In this case, a receptor kinase phosphorylates the cytosolic portions of the activated receptor. Once a receptor has been phosphorylated in this way, it binds with high affinity to an arrestin protein, which inactivates the receptor by preventing its interaction with G proteins. Arrestins also act as adapter proteins and recruit the phosphorylated receptors to clathrin-coated pits, from where the receptors are endocytosed. And afterwards, they can either be degraded in lysosomes or activate new signaling pathways.
After activating the alpha subunit by causing an exchange of GDP for GTP, remember the alpha subunit separated from the beta-gamma complex. We can then follow the beta-gamma complex, and one of the things it can do is travel along the cell membrane and interact with an ion channel. In this case, it's a potassium channel. We can see that the channel remains open and potassium can exit the cell when the beta-gamma subunit dissociates from the channel and reforms a complex with the alpha subunit. After the GTP has been hydrolyzed, the channel closes. So this is one of the effects that can come after turning on a G protein coupled receptor. Another common effect downstream of the G protein coupled receptor is production of a molecule called cyclic AMP. The text calls it a small signaling molecule, but it's also commonly called a second messenger. And we start here with ATP, and we can see that we're just modifying ATP, removing two phosphate groups, and then creating this ring structure that is cyclic AMP. So this is cyclic AMP. It's the active molecule we're going to be looking at in the next few slides. So what I've done here is, rather than using the pictures from your textbook, I'm also including them if you want them for reference. I'm going to try to show you how a pathway of a G protein coupled receptor can branch in many different ways. So in this one, I'm going to start with activation of the receptor, and we're looking at one ligand, adrenaline. Adrenaline is binding to the receptor. We have a conformational change. We're going to focus in on the alpha subunit, which is now going to release GDP and bind GTP. The activated alpha subunit is then going to interact with an enzyme called adenylyl cyclase. Adenylyl cyclase produces cyclic AMP from ATP. The cyclic AMP that is produced can travel throughout the cytoplasm of the cell. The cyclic AMP produced by adenylyl cyclase can then diffuse through the cytoplasm. And one of the things that can interact with is a protein called PKA, or protein kinase A. It's a protein kinase that's regulated by cyclic AMP, therefore giving it its name. When cyclic AMP binds to PKA, it's activated. And I'm going to start with here one branch of the pathway that happens downstream of PKA, one thing PKA can do next in a pathway. So one of the targets, one of the things that's phosphorylated by PKA is phosphorylase kinase. So we can see this is the inactive form of phosphorylase kinase that does not have a phosphate group. PKA is phosphorylating phosphorylase kinase. We get a phosphate group there. That's the active form. Now, phosphorylase kinase is not the effector. It's not the last part of the pathway. Phosphorylase kinase, see from its name, is a kinase, and it will phosphorylate its target, in this case, glycogen phosphorylase. We have the inactive glycogen phosphorylase and the active phosphorylated form. This is the effector. It is the protein that is going to cause glycogen breakdown. We'll look a little bit more at glycogen when we're thinking about the insulin signaling pathway, but glycogen is a storage molecule for glucose. So much of glucose is strung together as glycogen. To break it down, you just break the links between the glucose molecules, and those can be released to the cell. This is the image from your text showing you the same pathway I just described in my drawings. We're going to pick up in the next video, because I'm out of time, on how this pathway can diverge. You'll see how my drawings integrate multiple of these pathways into one drawing.